Yes, thank you very much. I would like to start by thanking the organizers for this opportunity to share our work with you. And as you'll see, it's a little bit of a change of pace because the focus of my talk is on the discovery of innate immune agonist. And at Spring, the approach we're taking to achieve this is to leverage advances in computation, specifically deep learning, and applying these to high dimensional phenotypic drug screens. I'm going to walk you through one of those phenotypic drug screens today. But first, just a quick introduction to our company. So Spring is actually broadly focused on age and age-related diseases. And we were founded on the principle that really to accelerate drug discovery in this complicated space, we're going to need to leverage things like machine learning really at every stage of the drug discovery process. What's unique about us as a company is we're pretty much an even split between wet lab biologists and computational experts. So we're teamed up early on with all of our experiments with a computational team, and they really inform experimental design and data interpretation. Now, with a focus on aging, we are specifically interested in the aging immune system. And a lot changes with age in terms of immunity, both on the innate side and the adaptive side. This all falls under the umbrella of immunosenescence. As a starting point, we've chosen to focus on innate immunity as the axis to generate therapies for many age-related diseases. So we actually have programs focused on decreasing inflammation. This is important as we age because basal inflammation actually increases, and this is often termed inflammaging. But you can also have very directed uses for decreasing inflammation. We have a clinical trial that just completed COVID-19, trying to reduce inflammation in severe COVID disease. We're also moving into other inflammatory diseases. But clearly today we want to talk about activating innate immunity and specifically activating therapeutic inflammation. And there's a variety of applications for innate immune activation in the context of aging and age-related diseases. And really the genesis of this project was a effort to discover new vaccine adjuvants. So it's well documented that as we age, our responses to vaccines decline. And it's also known that to improve these responses in the elderly, adjuvants are really key to this. Basically, by better engaging the innate immune system, by more robustly eliciting the right forms of inflammation, you can increase the likelihood that a vaccine will be protected in an older individual. Our focus is those small molecules. So anything we find in this vaccine adjuvant space, we believe can be translated into oncology, where you would use these compounds, as everyone else has today, to try to really change the immunosuppressive environment of the tumor. And then finally, I would like to highlight an indication here that dramatically increases as we age. It's actinic keratosis. This is the formation of neoplastic lesions on the skin. These lesions result from UV damage, and in many ways, they're the first step towards skin cancer, specifically cutaneous squamous cell carcinoma. And what's interesting about AK is that keratoses can be treated with innate immune agonists, particularly imiquimod applied topically. So we think there's opportunity here as well for innate immune agonists. So diving into the data types that we really focus on at Spring, there's two key ones. The first is high content imaging. So this is actually microscopy data, and I'll give more details about what we're doing here. And then also proteomics. We do this in the supernatants of our cell stimulation assays, and we're able to quantify quite a number of analytes. All of this information is fed into our computational analysis platform. And really the goal here is to construct what we term phenoprints. And a phenoprint is just a computational model. You can use these models to discriminate any number of things. They can be used to discriminate disease states. So you can actually discriminate healthy from disease cells using imaging and proteomics data. We can actually build models on aging, so we can determine whether a cell, such as PBMCs, come from old or young individuals using these data. And in this case, we're using or trying to build innate immune activation phenoprints. So stimulating cells with different activators of the innate immune system and building computational models that can discriminate these different forms of activation using this imaging data. So more on the imaging there. This is high throughput confocal microscopy. It's performed in a 384 well plate, and we capture multiple fields of view per each well. On the proteomics side, we're doing this at the level of the supernatant, so we're capturing information from the well. We have a B-based antibody capture system that we work with that enables us to analyze up to 200 analytes at a single time. For innate immune activation, clearly we're interested in cytokines, chemokines, and in some cases shed cell receptors. So there's much you can do with microscopy data, and really anything you can do with a confocal microscope, we can feed into our platform. But the core data set that we use is actually morphological staining. So this is often termed cell painting. So we use basically organelle-specific dyes that stain key organelles within the cells. So nucleus, cytoskeleton, mitochondria, cell membrane, Golgi, etc. And what you're trying to quantify are the morphological changes in your cells with stimulation conditions, and begin to understand sort of the physical and morphological associations that you have in these imaging data sets with your activation conditions. This has been well demonstrated using cell lines, particularly in drug discovery for cancer applications. But here we have decided to focus on primary immune cells. And we can do this with purified immune cells, but all the data I'm going to show you today is from unfractionated PBMCs. So we take PBMCs from many donors, plate them in our wells, and stain them with these morphological dyes. And it's from these images that we derive much of our data along with that proteomics information. There's a variety of reasons why this is beneficial. I'll just point out that this is highly scalable. So we do this in 384 well plates. It enables us to go through compound libraries consisting of tens of thousands of compounds. And in some cases, we build models using hundreds of donors. So we're able to generate these really large data sets to feed into our computational platform. So the screen I'm going to highlight today was performed on these unfractionated human PBMCs, multiple donors. These were stimulated with reference innate activators, which I'll go into in a moment, but basically control activators that target key TLRs or activate key forms of inflammation that we're interested in. And then finally, this is a screening campaign, so we have an experimental compound library as well. For us, this is a repurposing library. So these are compounds with annotated targets. So when we get a hit, we know what its primary target is. And also many of the drugs we are using are previously approved for other indications. So following that overnight stimulation of the PBMCs, you stain and acquire your imaging data, also collect the supernatants for cytokine and chemokine analyses, and all of this information goes into our computational platform. And what we do is we use this information to understand the relationship of our reference innate activators to each other, as well as to experimental compounds. And also because we're using microscopy data, you can actually dive into that data and begin to quantify automatically hundreds of features associated with the biology you're interested in. So for our reference innate immune activators, they come in two forms. As I said earlier, this was initially an adjuvant discovery campaign. So we actually utilize marketed vaccine adjuvants. What you really just need to know about these is that they're complex mixtures of multiple components. I'll call your attention to ASO3 and MF59. These are squalene oil and water emulsions. They're very similar in composition, and I'll come back to that in a moment. But we also do more targeted innate immune agonists. So you can do small molecules targeting TLRs, such as RH48, or individual PAMPs targeting really any TLR that you're interested in. We also focused on cGAMP as a stimulator for the Sting pathway. So you take your PBMCs, stimulate them with these reference compounds, plus the experimental library. And if you just begin to look at these images, you can almost convince yourself that you can begin to note differences here between the different reference innate immune activators. But what we're doing is we take all of this information from many donors, we're talking terabytes of data, and feed this into an algorithm. And it's going to begin to identify patterns for each stimulation condition. And using this imaging data, we are able to discriminate between conditions and begin to establish relationships between these different agonists and our experimental compounds. And so I'm going to walk you through this graph here because I know many may not be familiar with this, but this is a confusion matrix, and it's a way to show the accuracy of your computational models. So what we've done is we've taken PBMCs, we've stimulated them with these various conditions, and then we feed these images in machine, and it begins to recognize patterns for each individual stimulation condition. What we do is we leave out a subset of that imaging data, so we have images that the algorithm has never seen before. And once we are confident that the algorithm is robust and ready to go, we apply the algorithm to those previously unseen images and then test its accuracy. So the numbers here represent the predicted label, the label that the algorithm suspects these PBMCs were stimulated with these conditions versus the true label based on how we constructed the experiment. So if you start with DMSO, our control, you can see 951 times. The algorithm correctly called DMSO as DMSO with 99% accuracy, so quite good. Occasionally, there are a few misclassifications, RH48 and poly-IC in this case, misclassified as DMSO, but still overall a very high degree of accuracy. As you go through the conditions, you'll see that this accuracy is pretty much maintained across them. I'll highlight LPS and nigericin. This is a pretty inflammatory stimulus. It's actually going to induce quite a bit of pyroptosis hypothesis. In this case, we're able to construct a very accurate model. It's very distinct to the algorithm. I'll focus your attention again on those vaccine adjuvants, AS03 and MF59, because compositionally they're very similar. But what's interesting here is that the algorithm doesn't really confuse these things very often and is able to discriminate each with a high degree of accuracy. But it's not just enough to discriminate your different conditions. You want to begin to understand the associations of your reference conditions to experimental compounds. This here is a UMAP projection. It's somewhat similar to a t-SNE. So it's just a way of reducing high dimensional data down to 2 axes so that we can visually start to understand how different conditions are related to each other. I'll call your attention again to those squalene-based adjuvants, AS03 and MF59. You can see they're close together in the space, indicating their relationship, but again, quite distinct. The algorithm is able to distinguish them. As you move through this cloud, you'll see other vaccine adjuvants. We see that CGAMP is sprinkled in here with poly-IC on the end. We can focus on some TLR activators, R848 and LPS, still quite distinct clusters here, but closer together, they're in association. And then finally, CPG. This is CPGB. It's going to be activating B cells, always a quite distinct cluster. And the clusters I'm not highlighting here, these were actually approved actinic keratosis therapies. They're not innate immune agonists. And again, you can see they cluster together and are quite distinct from our innate immune activators. So the value of this approach comes when you apply your experimental compounds and put them into the space, because then for any experimental compound, you begin to understand how it is associated with these reference innate immune activators, and if in fact it's associated perhaps with multiple innate immune activators. So this really forms the basis of our screening technology. We use these unbiased models, which we often term signatures for various TLRs, Sting, et cetera. And then we couple this with these targeted features. I've already mentioned the proteomics analyses, but also diving into these images themselves, you can begin to really dissect the biology here and look at a variety of things. I don't have time to tell you about all of the things that we quantify, but I'll just give you a few high-level examples that you understand some of the additional things we associate. And it's really all of this information that goes into what we term a phenoprint, which is the model that we use to understand our experimental compounds and controls. So cell composition is a key feature for us. We're not staining with antibody specific for different cell subsets, but using morphology alone, we've been able to build classifiers that can clearly distinguish different cell subsets. Lymphocytes and monocytes in particular are quite easily distinguished, but you can get even finer than that. So within the myeloid department, you can see that monocytes, as they become activated, even with just this morphological stain, they're quite distinct based on their staining pattern. We can also see early signs of macrophage differentiation, and though dendritic cells are fairly rare in PBMCs, you do capture a few. Finally, death is often associated with innate immune activation, and fortunately for us, different forms of cell death have distinct morphologies, and we're able to capture and quantify these. So macrophages that are dying, perhaps on the verge of necrosis, also apoptotic cells and pycnotic cells as well. All of this information gets fed into a compositional metric, which allows you to see how your individual stimulation conditions or your experimental compounds are altering the composition of your PBMCs. The list goes on with targeted features, and really we can build out any number of things depending on what we're interested in for a particular screening campaign. With antigen discovery or with adjuvant discovery and innate immune activators, we were keen to look at antigen uptake. You can do this by providing fluorescent proteins or dextrans and actually quantify their localization within myeloid cells. Proteasomes are often seen with activated myeloid cells. These are actin-rich protrusions associated with cell motility and also antigen sampling. Mitochondria as well are quite rich in information. By quantifying fission and fusion events, you gain insight into cellular energetics and even the stress condition of your cells. And then finally, not as relevant to this overnight stimulation assay, but M1 and M2 macrophages have very distinct morphologies, and you can build very robust models to discriminate these macrophage populations. So finally, getting into the screen itself, again, this is a 10,000 compound library of repurposed drugs primarily with annotated targets. You screen those relative to your controls and look at the ways in which things alter your phenoprints. I don't have time to go into scoring, but we can build custom scores. Basically, you can weight things that you're most interested in and penalize things that you want to avoid. So if you're interested in the compound similar to his particular TLR agonist, but you don't want too much IL-6 production, for example, you can build a custom score to only surface those compounds. And then I'll show you just a little bit of orthogonal assay data where we've tried to translate some of the hits we're seeing in the screen. So first, as a sanity check, it is an annotated compound library. So there were already annotated TLR agonists within the library that went in as experimental compounds. Within the library itself, there were 7 annotated TLR7 or TLR7/8 agonists, and we actually flagged 6 of these 7s in the screen for their similarity to R848. And this is just a snapshot from our internal software we use to sift through all of the screening data. It's just showing a very small subset of information, but you can see the annotated TLR agonists flagged here. And again, 6 of those 7 received a high or significant TLR7/8 score. We also have a custom scoring system that's based on weighting a variety of features that we're interested in. And you can see by using this, we were able to capture all of these TLR agonists. But we identified a variety of other compounds with similarities to our controls and have started to now translate those into orthogonal assays. So in many ways, one of the simplest assays to look for the ability of these compounds to engage the innate immune system is to look at dendritic cell maturation. We are particularly interested in this from a vaccine standpoint, but clearly it has implications for oncology applications as well. And so these were hits taken from the screen. We applied them or used them to stimulate monocyte-derived human dendritic cells for 24 hours, and then just looked at their ability to upregulate maturation markers on DCs. I'm showing a FACS plot for CD86 here. So you can see the DCs, they are differentiated. They do express CD86. As we add on some positive controls, here LPS or poly-IC, that CD86 goes up. And then I'm highlighting 2 drugs, which unfortunately I can't share with you the targets today, but both of these behave similarly to poly-IC here in terms of the magnitude and degree of CD86 upregulation. And what's interesting about drug 1 and drug 2 is that they were flagged for high similarity to a vaccine adjuvant, ASO3 in this case. They also induce quite a bit of cytokine production, particularly IL-1 beta production. They do this in both PBMCs as well as dendritic cells. And they were originally developed as cancer chemotherapies. So these drugs were developed to be cytotoxic, but what we're seeing in the system is that you can take these down to concentrations where they're not inducing cell death and they're still eliciting forms of inflammation similar to the controls that they were flagged for. Encouraged by this initial result, we decided to take one of these drugs and take it all the way into an animal vaccination model. So this is drug number 2, and this is just a mouse model of influenza vaccination. It's a recombinant protein basically injected into the muscles of these mice. You prime on day 0, you boost on day 14, and then you bleed on day 28 to look for specific antibodies to nucleoprotein. And just to walk you quickly through the data, protein antigen alone gives you a mild response. An adjuvant, in this case, the control was alum. It's most frequently used in this model. This is a fully formulated adjuvant, gave a nice antibody response. But you can see that using drug number 2 here, we were able to recapitulate this and at least perform comparably to alum, suggesting that drug number 2 is to trigger DCs, as shown in the previous assay, and also functioning as a vaccine adjuvant in this context. And what I would say is this drug has not been formulated in any particular way other than put into PBS and combined with this protein. And so we're undertaking studies now to really refine the formulation for this to try to improve efficacy even further. And then finally, the last little bit of orthogonal data that I'll leave you with is some of the early translation we're starting to do into syngeneic tumor models. One of the things we've noticed is that several key classes of chemotherapies were flagged in our system for inducing inflammation similar to a lot of our controls, in particular, in this case, some of the adjuvant controls. What's interesting about these drugs is as they were initially being developed, they were primarily put into xenograft models, so animals without an intact immune system. And so we've undertaken some early studies to plug these into syngeneic tumor models and are seeing some early activity there. You know, I want to be clear, these are chemotherapies. We certainly appreciate that they're having cytotoxic effects as well, but based on the other data that we have from both the screen and the orthogonal assays with MODC maturation, we believe they also have independent and direct abilities to engage the innate immune system. And so we believe the screen is preferentially surfacing these chemotherapies that sort of have this dual role, this ability to directly target and kill cancer cells, but also to engage what may be desirable forms of inflammation. And we're conducting the studies now to look at the way they alter the tumor microenvironment, lead to perhaps infiltration with immune cells. And we're very keen to combine these drugs with checkpoint to see if we can preferentially identify chemotherapies that may combine well with checkpoint blockade. So in conclusion, just at a high level, we apply computational methods to high content imaging And we're using this approach to really build models to discriminate different forms of innate immune activation. We pair these models with proteomics analyses as well as targeted features within the images themselves, and use this to build a deeper understanding of our compounds, both the reference innate immune activators and our experimental compounds. And we believe we've shown early signs of pulling out some interesting compounds from this repurposing library that have potential applications as vaccine adjuvants or immunotherapies. And we're certainly interested in expanding this. I think one of the things I would love to see is a structure-based library where we use a system like this to rank something like a group of TLR agonists, things that have been structurally designed to have that form of engagement. I don't have time to thank everybody on the team, but this was a group effort. And with that, I will say thank you and take any questions.
Thank you. That was very good and bang on time. I'll just take the last question I have from the floor first. Are your mouse models on aged mice?
Yes, that's a good question. So the studies I showed today were all with young mice, but we actually do have models built on aging mice. So I focused on our ex vivo analyses, but we do a lot of work with histology as well. And so we actually have tissue aging models. So by examining various mouse tissues, we can actually predict age and screen for drugs to rejuvenate those.
Okay, and have you considered including epigenetic data as part of your analysis? Specifically DNA methylation?
Yes, yeah, absolutely. Epigenetics can be a great predictor of age in particular. We haven't undertaken any studies to combine that with our phenotypic approach here, but that's definitely something we're considering.
And another question, CGAMP stimulation was not recognized. Seems to be an obvious exception. Can you comment why?
Yeah, that's a great pickup on the data there. We haven't tried other Sting agonists, but you're absolutely correct. I do wonder if I use something to permeabilize the cells, perhaps get a higher concentration intracellularly, it might work better. But I would be interested to try other activators of Sting to see if they have a more pronounced phenotype.
And then another question, I was curious why the Sting agonist had a much lower prediction percent from the PBMC imaging data, similar to the above question by Josh.
Yeah, absolutely. No, it's a great question, and we're still conducting experiments to deconvolute that.
Okay, and what are the different cell death pathways that you screen for? And was there any correlation with specific cytokine release, inflammatory markers, etc.?
Yes, absolutely. So we do have a pretty big focus on pyroptosis, are able to quantify that. So clearly that's going to be associated with IL-1 beta release. I would say that and apoptosis, you're really thinking about different cell modalities that have clear morphologies. Those are pretty clear to quantify in the data.
I'll take one last question. How many PBMC donors were used in your study? Were they individuals or pooled?
Yeah, so we use individual donors, but we do run many replicates. For a typical screening campaign, we usually start with around a dozen or so donors. But for some of our finer models, especially when we build aging models, we have built models to look at the ways in which age affects innate immune activation. There we get as high as 100 donors per each model.
Thank you very much.